Hello students. So welcome back to the channel. In this video, we are moving further and we'll be discussing about chemiosmotic hypothesis. Now see, chloroplast and mitochondria, they both will generate ATP by a method that is known as chemiosmosis, which is proposed by Peter Mitchell. Chemiosmosis, what does it require? It requires one membrane, it requires proton pump, proton gradient, and ATP synthase. Now see, ATP synthase, it has got two parts. One is CF0, which is embedded in the thylakoid membrane, and it makes a channel so that facilitated diffusion of the proton can occur. Reason is proton, which is a charged particle, is not able to cross the membrane. So they want the help of a protein. So F0 will act as a transmembrane channel for facilitated diffusion. Is it clear? Another portion is CF1, which is coming out on the outer surface, and that will be the area where, because of the movement of the proton, conformational change will be there, and ADP plus inorganic phosphate will combine to form ATP. So see here, this is the way how the work is done. In the step 1, ADP plus inorganic phosphate will combine here, then rotation. Step 2, ATP is made, and finally in the step 3, ATP is released. Now see, synthesis of ATP, whether in the mitochondria and chloroplast, both are using electrical potential energy to generate ATP. Now what is electrical potential energy means? Electrochemical gradient. In the electron transport chain, which is working both in mitochondria and chloroplast, they are accumulating proton and they are creating a gradient. Difference is in mitochondria, the proton is accumulated in the intermembrane space, and in chloroplast, proton is accumulated in the thyroid. Check beta this diagram, mitochondria and chloroplast. So if you see here in the mitochondria, chloroplast is accumulated in the intermembrane space when the electron transport chain is working, and in the chloroplast, it is accumulated in the thylakoid space. Is it clear? And this proton, when it comes back, because we have to break down the gradient, it is coming back. It is coming in the stroma in chloroplast, and it is coming in the matrix in mitochondria. ADP plus inorganic phosphate is combining to form ATP. So this diagram is clear. So electron transport chain will transform redox energy to pump proton across a membrane to create a gradient. And it is stored as a difference in the hydrogen ion concentration. And this, why? Because we know membrane is impermeable to protons. Now, why we are interested in creating a proton gradient? See, this gradient is important because when we break this gradient, then only ATP will be formed. So protons are allowed to move along the gradient, but because they are charged particle, their diffusion is facilitated, and that protein which is helping in diffusion, that is your CF0, CF1 in chloroplast and F0, F1 in mitochondria. So gradient is broken down by the movement of proton across the membrane, and they are coming to the stroma through the transmembrane channel. Is it clear? And breakdown of the gradient will provide energy to bring the change in the F1 where the ATP synthase is present. So you can see here, this is either thylakoid membrane you say, or you say inner membrane of mitochondria where your F0, F1 is present, or CF0, CF1 is present. Here higher concentration of proton. This membrane is impermeable to proton. And when the proton are moving, that time ADP is combining with an organic phosphate to make ATP. And movement is from the high concentration to low concentration, which is matrix in mitochondria or stroma in chloroplast. This is a very basic, we should know why we are interested in creating proton gradient. Because then only we can make ATP. Now let us see how chloroplast is creating proton gradient. So first of all, you know, splitting of water molecule, analyze the diagram, splitting of water molecule is occurring on the inner side of thylakoid membrane. Oxygen is liberated and proton is accumulated in the lumen. Now from the electron acceptor, it is given to the plastoquinone. Understand, beta, plastoquinone is not an electron carrier, it is a hydrogen carrier. It is clear? It is a hydrogen carrier, so it is removing the proton from the stroma and it is becoming reduced to form plastoquinol. But when it passes the electron to next protein complex, that is cytochrome B6F, the proton is separated from the electron and proton is released. So plastoquinone is removing and accumulating. Photolysis of water is only accumulating. And from the plastocyanin, it is given to PS1. And finally, it is given to the NADP+, which is removing the proton from the stroma to become NADPH. So in this diagram, two area proton is accumulated, one and the PS1, 2 during the photolysis of water, one by the plastoquinone. Is it clear? Two area protons are accumulated, one by PS2 and one by plastoquinone. Two area proton is removed. One is by plastoquinone is removed and one is by the NADP+. So what is the result? In the stroma, proton is low, but in the lumen, proton is high, and we are creating electrochemical potential gradient. Clear, beta? This gradient is because of the charged particles and it is because of chemicals. So we have created a gradient. And now this proton is passing from CF0. This is CF0 particle. And this is CF1. Already I have explained you in the previous, right now only, previous slide. So ATP synthesis is done. So I hope so this diagram is clear. Just see the theory also once again. Splitting of water molecule will take place on inner side. So protons are accumulated. Then electron is moving to the electron transport chain. And what happened? The primary acceptor of electron, which is located towards the outer side of membrane, the primary acceptor is pheophytin, will transfer its electron not to an electron carrier, but to a hydrogen carrier. And who is hydrogen carrier? Here is your plastoquinone. Clear, beta? So this molecule will remove the proton from the stroma and it will be pumping it into the lumen. So when this molecule passes the electron to the electron carrier, proton is accumulated in the lumen. So plastoquinone is removing plus accumulating. Photolysis of water is only accumulating. Clear, but it is only accumulating. And NADP reductase enzyme, which is located on the stroma side, it is actually removing the proton from the stroma. So final, what is the conclusion? Inside the chloroplast, proton is decreasing in the stroma, but increasing in the lumen. So this creates a proton gradient and we have to break this gradient to make ATP. Up till here, clear? Another question based on this diagram is, if you see plastocyanin is present on this side of the membrane. So plastocyanin is hydrophilic molecule because it is facing the head of the phospholipid bilayer. But plastoquinone is embedded in the tail. So plastoquinone is hydrophobic. Let me show you the next diagram very clearly. See here, beta. This is your photosystem 2. Photolysis of water is accumulating proton. Plastoquinone, you can analyze the diagram. It is within the tail, means hydrophobic. It is removing the proton from the stroma and accumulating in the lumen. Then your cytochrome complex giving to plastocyanin, which is facing the head. So it is hydrophilic, giving it to the PS1, which is finally giving it to the enzyme, this enzyme, which is removing the proton. So here what is going on? Proton gradient is created. Inside this thylakoid high proton is there. Stroma has low proton, so it is coming and making ATP. So ATP is made, NADPH is made, we'll be using in the Kelvin cycle. One more diagram just for your more understanding. See this. I hope so this diagram is easy enough. Plastoquinone is hydrophobic and plastocyanin is hydrophilic. Clear? This is one protein complex. This is another protein complex and this is one. So in the previous video, we have already studied the light reaction involve many protein complex. One is your photosystem 2, then cytochromes, and then photosystem 1. And this is your F0 and F1 particle. In the chloroplast, we will use word C. So I hope so. In this video, I tried to explain you chemiosmosis. It might be easy for you. Thanks for watching.